I'd like to, can everyone hear me in the back? I'd like to thank the organizers, and particularly Professor Nagi Habib, one of the organizers, kindly providing this invitation for me to present my work today. I just moved to University of Miami last year, so I was at UCLA for 10 years before that. And like Professor Habib, I was involved in the, Professor Habib's also past president of the International Society of Cell and Gene Therapy of Cancer. So I have to disclose first that I'm a consultant for a company called Tokagen, which has developed the technology, is commercializing some of the technology you'll hear about today. Just by way of introduction as background, so cancer, as you've just heard, is certainly a target disease for gene therapy. And in fact, even in the first decade of gene therapy, clinical trials for cancer far outstripped all other indications. However, at the same time, it was recognized by the, this is the Orkin-Motulsky report commissioned by Harold Varmus when he was NIH director to try to assess the NIH investment on gene therapy research. And here, at the same time as cancer was becoming a major target, it was recognized that, as you can see from the conclusions here, major difficulties at the basic level include shortcomings in all current gene transfer vectors. So this was a problem, and the problem is exemplified here. This is a section through a human brain tumor, a glioma, treated with AD5, a conventional adenovirus serotype 5 vector, a non-replicating vector, because in the early days of gene therapy, of course, the watchword was safety. So all the vectors had to be rendered replication defective. So this is an AD5 vector expressing P53, and you can see that the P53 staining only extends for a small distance around the needle track, and the majority of the bulk of the tumor is untouched. So conventional non-replicating vectors, even adenoviral vectors at very high titers, do not achieve effective levels of intratumoral gene transfer. They are diffusion limited, and the high hydrostatic pressure in the tumor prevents diffusion and effective penetration. So, and in clinical trials for glioblastoma, for example, which went to phase 3 in the early days of gene therapy, despite prior debulking and multiple site injections around the tumor margin, it was found that the overall tumor transduction efficiency was less than 1% and certainly therapeutically inadequate. In the last 10, 15 years, there's been increasing interest in using replicating viruses as cancer therapeutics. Because, of course, if it's a replicating virus, it does what viruses do best, which is spread from cell to cell and make more copies of themselves. And then there's the potential to achieve better tumor transduction and penetration. So, and in fact, it would be even better if you used another attribute of viruses, which is that they many of them lyse their hosts. So the reasoning is that if you could develop an so-called oncolytic virus, which does not replicate or is inhibited from replicating in normal cells, thereby sparing them. However, within cancer cells, selectively replicates and bursts out of its host, then you might have an even better effect. And in fact, there are all of these viruses now are currently in clinical development and being evaluated in clinical trials as cancer therapeutics. And this heralds the return of a field that used to be called viral therapy. And viral therapy actually had its heyday in the 1940s, 50s, where there were extensive clinical trials based on case reports dating back even to 1901 of patients like this who had, for example, this child had a lymphoma, Burkitt's lymphoma, and you can see that after a spontaneous measles infection, this was a case report in Lancet in the 1970s, showing that there was spontaneous remission of that measles infection, suggesting that oncolytic viral therapy might be able to work. We now understand that the mechanisms underlying the tumor selectivity of viral therapy are real, that basically because we now know that tumors are extremely immunosuppressive environments where not only adaptive immunity, T cells and B cell immunity are suppressed, but also there are cell intrinsic defects in innate immunity and interferon signaling, which basically signal antiviral defenses. These are all suppressed, and this makes, or mutated in tumors, and this makes them a uniquely susceptible, permissive niche for viral infection. And so basically, that is the reason for the success of, or partial success of oncolytic viral therapies. This was one of the first oncolytic viral therapies developed about 10 years ago now. This patient slide was kindly provided by David Crowne, who was at Onyx at the time, for a review article that I wrote. But basically, Onyx 015, this is a patient with head and neck cancer who had failed prior cisplatin 5-FU. This is a recurrent tumor, head and neck cancer. And you can see that after some cycles of treatment with this oncolytic adenovirus, which is one of the prototypic virus, you can see that the tumor regresses. If you biopsy the regressing tumor, you can see within the nuclei, you can see almost crystalline arrays of packed viral particles that are replicating in the tumor. Now, this seems like quite a good therapy and successful. In fact, this type of therapy was approved in China as one of the world's first gene therapy modalities to be approved in the world. However, in this particular patient, for example, the tumor recurred about 6 months later. And basically, this is and the virus, however, is cleared. So this is the conundrum of viral therapy, that basically, as these lytic viruses, they lyse cells as they go, they burst out of the cells, they kill their host cell, they kill and destroy the permissive protective environment of the tumor, which allows them to persist, and therefore open themselves to the immune system. And therefore, and the immune system, of course, even with adenovirus, even with non-replicating vectors, we know from the gene therapy field that they're very potent humoral and cellular mechanisms to clear viral infection. So it becomes a race between the virus trying to piecemeal kill cells as it spreads through the tumor, and the tumor protective environment being destroyed, and a race between the virus and the antiviral immune response. So half of the field in this currently is trying to suppress the immune response to enhance the viral effect, and the other half noted that there is an anti-tumor response that also is engendered at times, and so is seeking to augment the immune response to try to seek to harness that anti-tumor response. So it's a delicate balancing act. So some years ago, we basically thought to take a different approach, which is not to use viruses. All the viruses in that previous list that I showed, including exemplified by adenovirus, which is the prototype, are extremely high titer cytotoxic cell-damaging viruses. So these viruses have the lifestyle you're familiar with, which is to make as many copies of yourself as you can, and then burst out of the cell. So then move on. So destroy, pillage, move on. Those types of viruses don't integrate. They have no need to integrate into the genome of their cell. However, they're extremely immunogenic because they raise all these danger signals. However, retroviruses, as you know, are permanently integrating viruses. They are actually the flip side of the coin. They're benign parasites which integrate permanently into the genome of the host cell, and therefore every daughter cell of every infected cancer cell, every daughter cell will have a copy of the virus in its genome. It cannot get rid of it, and basically this allows extremely long-term tumor persistence. In particular, murine leukemia virus, for example, is a simple retrovirus which only infects dividing cells. And so it has no nuclear localizing signals in its capsid, and therefore has an absolute selectivity for rapidly dividing cells. So we found that these viruses are extremely selective for cancer. In the same way as other oncolytic viruses, they can spread in the immunoprivileged tumor environment selectively. And again, they only infect proliferating cells. Because they're an integrating virus, they don't kill cells immediately upon infection, but spread rather stealthily. And then you can, even though they're not intrinsically cytolytic, you can outfit them engineer them to have a prodrug activator enzyme, a converting enzyme, which activates a non-toxic prodrug into a cytotoxin directly in the infected cells. So this allows long-term control of cancer because you can't infect every single cell, at least not at the outset. But because the virus stably integrates into the tumor, this creates a viral reservoir which will reinfect tumors even as they recur. So some of my students basically showed previously that you can take primary glioblastoma multiforme. This is the most aggressive grade 4 type of primary brain tumor. And you can see that the virus with an EMD is emerald green fluorescent protein shows very nice replication kinetics on the majority of these primary patient-derived isolates. So instead of a marker gene, we can now outfit this with an internal ribosome entry site driving cytosine deaminase from yeast, which is turns out to be a prodrug converting enzyme for 5-fluorocytosine. 5-fluorocytosine is an antifungal agent which works on yeast, but its mechanism of action is that it's actually converted to 5-fluorouracil, a classic anti-cancer drug, by this enzyme, cytosine deaminase. So this is essentially a prodrug converting enzyme, also known as a suicide gene. And it turns out that 5-FC, the antifungal agent, which serves as the prodrug, actually crosses the blood-brain barrier and is in fact FDA approved for CNS fungus infections as a drug in its own right. So some years ago, Chen-Kuo Tai in my lab set about to do this experiment where he injected U87 human glioblastoma cells into an animal intracranially, established a tumor. He put in over tenfold less virus to try to make it a realistic experiment, and then asked for virus to spread, and then gave the prodrug in weekly cycles. And you can see that compared to the control groups, which received only virus or only prodrug or PBS, you can see that the PBS group, you see multiple tumor nodules developing as from the initial inoculation site as the tumor cells migrate through the CNS. But then after one shot of virus, a single shot of virus, you can see all of those tumor sites are marked by immunohistochemistry for viral proteins. And you can see that the virus basically tracks the tumor as it spreads through as the tumor cells spread through the CNS, because it now is permanently part of the tumor cell genome and spreads through to other daughter cells. Now, if you give virus and then you give multiple cycles of prodrug, you can see that the median survival has enhanced tremendously, and you can see that most of those lesions are now eradicated. If you track this by luciferase, you can see that tumors in intracranial tumors in the control groups grow inexorably and cause death of the animal. But with each prodrug cycle, shown by the shaded areas here, you can knock down the luciferase signal. So in real time, you can see that you are treating the tumor and you're getting knockdown. Now, this can basically achieve long-term tumor control, but we never fully see complete loss of the signal. So we don't see eradication in this xenograft model of human glioma in an immunodeficient animal. Now, if you do this in an immunocompetent animal, now you see a different story. You see that within 2 or 3 prodrug cycles, the tumor signal goes down and stays down. And you can take them off prodrug, and they will continue to survive for over 300 days in this experiment. And if you see, and again, this tumor, the control groups all die. However, the treated group, even after only 3 prodrug cycles, show full eradication. And we see that, in fact, if you take those animals which lived over 300 days, and then you re-challenge them subcutaneously, now with naive tumor cells, non-infected tumor cells, parental tumor cells, then you can see that you see the control animals, which never saw tumor before, show engraftment of tumor. However, the previously cured animals now reject the tumor, indicating a memory effector response, which we can also document through the use of ELISpot. So we see that this is actually a twofold effect. We see that you can have viral spread, stealth spread throughout the tumor, which does not initially damage the tumor cells. Once you get maximal spread, you can pull the trigger on all the cells at the same time by giving the prodrug. You can regress much of the tumor. In animal models, we've seen 99% transduction of tumor or better, but you still will have that 0.01% that will recur. And you can do that for many cycles, but eventually you'll get the immune system activated against the cancer. And you can see these are patient samples, which actually I'll show again later, but documenting that support for our hypothesized mechanism of action. And we see in lymphocytic infiltration, and you can see that the immune system is activated against the cancer. So our first clinical target was indeed primary recurrent brain cancer, high-grade glioma, both glioblastoma multiforme as well as anaplastic astrocytoma. And the median survival after newly diagnosed patients is 10 months. With surgery, radiation, alkylating agent, chemotherapy, you basically have maybe a 16-month survival prognosis. However, once you recur, you are now temozolomide resistant and perhaps radio resistant, and the median survival after recurrence is about 8 months or 32 weeks. So this is a dire prognosis. So in 2009, we pulled together an investigational new drug application in collaboration with a newly founded biotech company, Tokagen, and this was a 3,000-page filing because FDA had never seen a live retrovirus being proposed for use in humans before. So this second generation, we had improved the viral vector, developed clinical grade manufacturing procedures, and in mid-2010, we did our first in-human injection of this product, which is now called TOCA 511, a retroviral replicating vector in this 34-year-old patient with recurrent glioblastoma, conducted by Professor Linda Liao, Vice Chair of Neurosurgery at UCLA, where I was at the time. And so we see, since then, we've treated over 90 patients to date in 3 clinical trials total. We've seen minimal adverse events. Throughout all those trials, we've seen only grade 1, 2 adverse events for the most part, which are fever, etc. We've seen only 1 grade 3, 4 with relation to which is probably not related to the product, which is asthenia in a patient who already had a pulmonary embolism. And we've seen some leakage indicating some weeks later after viral injection, we've seen some blood signals, but you can see that once it leaks out of the permissive environment of the tumor, neutralizing antibody titers climb, and you can see that the RNA signals in the blood just completely disappear. So outside the permissive environment of the tumor, this virus is indeed cleared and well controlled. So we have now conducted 3 clinical studies, over 90 patients treated as of this June. One is a direct MRI-guided intratumoral injection, stereotactic intratumoral injection study. Another study that we started in hopes of improving the physical delivery of the vector was a resection study. Here you resect the tumor first and then inject into the tumor margin cavity walls to try to get residual tumor. And then just last year, FDA allowed us to initiate an intravenous trial. So the results of the intratumoral study are pretty much same as the resection study, but you can see that even in early patients with low dose cohorts, these are dose escalation phase 1 trials, even at low dose cohorts, we found that beyond the injection site, you could see by qPCR, that's what these pluses indicate, you could see positive signals by qPCR even out to the margin of the tumor after scheduled resection. And if you stain, you can see immunohistochemically anti-GAG antibody indicating viral protein production. And whereas you see the selectivity in adjacent brain tissue, you see no such signals. After convection-enhanced delivery, for example, at the UCSF trial site, so we now have 7 or more clinical trial sites across the United States. After convection-enhanced delivery into this large lesion, you can see that after even a single cycle of 5-FC, you see a great reduction of the enhancing area. And we see very similar results in the resection study. And here, again, you can see extensive necrosis in tumors. Now, this was a tumor, interestingly, that radiographically actually increased in size, or appeared to increase in size during treatment with the prodrug. And this appears to be a case of pseudoprogression, where instead of actual tumor progression, which is what you normally would think it is actually is a case where the tumor is dying, and lymphocytes are coming in and packing into the tumor, and you're seeing this kind of swelling and edema, which suggests progression, but is actually now known to be pseudoprogression. So when you actually take the tumor out, you see extensive necrosis, you see dying cells, and you can see in the residual cells, you see the virus is still persisting. And again, in these patients, you can see that we get radiographically shrinkage of the tumor. And even in uninjected lesions such as this, we can get complete responses showing that you're getting a distant site effect, suggesting an immune response. So overall, across all dose cohorts, even the very lowest dose cohorts, which we thought would be subtherapeutic, we see compared to the historical benchmark of 32 weeks, 8 months for recurrent high-grade glioma, we see in the intratumoral study 53 weeks, in the resection study 52 weeks of overall median survival, overall survival, suggesting that we have a significant improvement compared to historical data. And in particular, when you subset out the grade 4 glioblastoma patients, that's 61 weeks. The OS6, the overall survival at 6 months, is 94% now compared to historical benchmarks of about 59%. And progression-free survival also follows similarly to overall survival. And you can see that compared to progression-free survival historical benchmarks, which are about PFS6 is about 10%, median progression-free survival is 9 weeks. You can see the progression-free survival is about 26 to 35%, with median progression-free survival 14 to 15 weeks. So, and most encouragingly, as we go out further, after 20 or 30 weeks or so, we see this tail where no one's dying or progressing anymore, which basically suggests that we have indeed invoked an immune response which gives you long-term anti-cancer immunity. So finally, we have, just last year, the FDA felt confident enough with our safety data in patients to allow us to initiate an intravenous study. And this was based on studies that were performed in collaboration with Tokagen, showing that if you give high enough doses of vector IV, that you will go through the leaky neovasculature of the tumor, and the virus will be retained and can reach effective transduction levels in the tumor. And this is the data now, pharmacokinetic data from our first patient cohorts. These were initially single patient dose cohorts, 301, 302, 303. Each had 10-fold increases in their vector, or 5-fold increases in their vector dose. And then from patient 3, it converted to a 3x3 dose escalation. But you can see that these are the first 2 patients. This is the third patient in green. You can see that at this dose of 10 to the 9 or so, we now are seeing effective, much better pharmacokinetics in the tumor and in the blood. So in summary, this retroviral replicating vector-based approach appears to be safe and well tolerated across all dose cohorts. We see successful transgene delivery in terms of both viral proteins and the therapeutic payload, cytosine deaminase suicide gene protein. We see tumor necrosis after giving the prodrug. We see preliminary survival data that surpasses benchmarks across all cohorts, and we see that we're likely to be generating 5-FU directly within the tumor. So this is a form of intracellular chemotherapy, if you will, where you're forcing the tumor cells to create their own chemotherapy, which now leaves you an intact immune system because you have no systemic myelotoxicity. So now your immune system does have the capability to come in and clear the tumor and benefit from immunotherapeutic approaches as well. Now that the FDA has allowed us to do an intravenous trial using intravenous delivery of the vector for brain tumor patients, this opens this to approach to other cancers, systemic cancers as well. So some years ago, Kei Hiroka in my lab showed that you can do local regional infusion through the portal system and get this is a multifocal metastasis model. These are actually livers, even though they don't look like it, which are studied with multifocal colorectal cancer metastasis in the CT26 syngeneic model. model. And you can see that you get, at the research grade level doses, titers that we could achieve in the lab, you get most of the tumor foci, but not all of them. And again, you see this exquisite selectivity for the tumor as compared to the normal liver parenchyma. Now, Tokagen has now used clinical grade high titer virus, which is about 1,000-fold higher than we could achieve in the lab. And now you can see that this is F-Luc, luciferase marked tumor foci, and this is the GFP viral vector with the reporter gene. And now it may be hard for people in the back, but basically if you look at these tumor foci, every single tumor foci has virus in it, and which is in the process of spreading. So at high enough titers intravenously, you can get more successful tumor transduction. This is important to us because in vitro and in vivo models, by direct intratumoral injection, we've seen efficacy supported in many cancer types, not only GBM and colorectal cancer, but prostate cancer, breast cancer, bladder cancer, and others. So Tokagen's pipeline currently is to, we are initiating a phase 2b/3 registration study as of September this year, which will be conducted in 50 clinical trial sites across the United States, involving 170 patients. And we will seek to get approval for the treatment through that study. We're also going to try to go after other indications such as colorectal cancer and metastatic melanoma. This is a broad-based platform. You can put in various therapeutic genes, other prodrug activator genes, as well as RNAi sequences, for example, to suppress immune checkpoints, as well as cytokines. And we've shown that you could deliver all of these, at least in research papers. So I'd like to just acknowledge the people in my group who've done this work. Chris Logg was a former student and then eventually became my assistant professor at UCLA, who did a lot of the pioneering work on this. I'd like to acknowledge also Sarah Collins and Aki Inagaki, who are now came with me to University of Miami and who are pursuing these studies as well. I'd like to acknowledge many collaborators in various, including Professor Habib and various collaborators both in the United States and Europe who've helped in the development, preclinical development. And I'd like to most particularly thank the Tokagen management team, who are extremely professional. Harry Gruber and Doug Jolly basically started one of the first gene therapy companies in the world, Viagene, which was then subsequently bought by Chiron in the early days. And then Harry became a serial entrepreneur and came back to the gene therapy field recently with this company. We just hired Jamie Skillings. She conducted the phase 3 trials for Erbitux at Bristol Myers, for Avastin at Genentech, and for Xalkori at Pfizer. She's an extremely talented phase 3 trialist, and we're very fortunate that she's conducting our phase 2b/3 trial. And I'd finally like to acknowledge our clinical collaborators at the various sites across the United States: UCLA, UCSF, San Diego, Cleveland Clinic, Henry Ford, JFK Medical Center, Ohio State, and Swedish Institute in Seattle. And thank you very much for your attention.
We have quite a few minutes for questions, please.
Yes. Oh, no. Questions? Yes, please.
I'm very interested in this work.
Thank you.
I'm just wondering how much flucytosine are you giving to the patients, and have you looked at the sort of levels of 5-FU that are generated by flucytosine compared with conventional 5-FU administration? And the reason I ask this is that I do question how much of the efficacy you're seeing is due to the production of 5-FU. And many years ago, we looked at
Certainly, yeah, that's a very good point. First of all, this is, unlike all previous generations of conventional gene therapy vectors, this is a replicating virus. And therefore, again, in the early days of gene therapy, even in clinical trials, we found that actual transduction efficiency within tumors was about 1% if you used a non-replicating virus. Here we're at about 99+%. At least in the animal models. And in humans with dose escalation, we are seeing higher and higher levels. In some tumors, obviously human tumors are much bigger than animal tumors, animal models, but in human tumors, in some regions, even in the early dose cohorts where we were able to get resection specimens, we have at least 60+% transduction levels. And so I think we are able to generate much more 5FU than traditional gene therapy models would predict. I think that the gene itself is actually a much better version of cytosine deaminase than also was previously used in most gene therapy studies, which usually used E. coli cytosine deaminase. And this is yeast cytosine deaminase. And it's also human codon optimized and thermostabilized so that instead of a room temperature optimum, which yeast-like, it's at the thermal stability is much better at 37 degrees, which allows it to, we've calculated, we've measured bioactivity is at least threefold better on a per molecule basis for generating 5FU. I think that the 5FU is particularly in the brain. In the context of colorectal cancer, certainly you can infuse a lot and it'll get into the tumor sites systemically, but you also kill the bone marrow in that sense. In the brain, 5FU fails to penetrate the blood-brain barrier utterly, and so has never been used as an agent in brain tumor, in glioma therapy classically. However, we showed that you can, by giving 5FC and converting into 5FU, you can get very, very effective killing of glioma cells in vitro and also in these cancer models in vivo. And again, 5FC, for some reason, does pass the blood-brain barrier, unlike 5FU. It seems that there's a CNT transporter protein in the blood-brain barrier that allows selective penetration of cytosine and not uracil. Thanks. Yes?
Can you elaborate a little more on the long-term survival and the immunological response in those long-term survival after removal of the anti-drug treatment?
Yes. These are still interim data. The studies are still ongoing, and so we're still assessing the long-term survival. But this has been across all dose cohorts. If you do a swimmer's graph, we see, which is basically kind of the survivorship of individual patients. We see that it's not dose dependent. It's not dose cohort. Necessarily dose dependent. In some patients, even in the earliest dose cohorts, in fact, one of our longest living patients, which we followed the longest because he was in the earliest dose cohort, has lived more than 3.5 years. We see a considerable percentage of patients who are living for more than 1 to 2 years. We see a trend towards better survival in the higher dose cohorts, which were started later. But those patients are shorter in follow-up. But encouragingly, what we are seeing is that with every month that goes by, we're seeing the data are maturing and they're actually improving for the higher dose cohorts. So we do think that in the end, we will see a dose-dependent effect, viral dose that correlates with longer-term survival. Yes?
What is the mechanism? That's
the immunological mechanism. We haven't been able to do a lot of immune monitoring for these patients. We do see histological evidence, as you saw, of lymphocytic infiltration, but a lot of tumors may show some lymphocytic infiltration. We see a lot in areas of necrosis immunohistochemically. We have not done, and this is something that we're planning to do in the phase 2b trial, to actually take peripheral blood from the patients and analyze to see whether they have anti-tumor responses. But this is not part of the original phase 1 protocol, and so we weren't able to consistently do this. But we do think that there is a strong immunological component based on the preclinical data and the very different results we get from the immunodeficient models versus the immunocompetent models. Yes?
Yeah, very interesting talk. But in the syngeneic models, where you saw the reintroduction of the tumor cells back into those animals and eradication of those
Yes.
tumors as well, that would suggest that you're seeing tumor epitopes rather than viral epitopes. that are
Yes.
being there. Is there any evidence, although you just mentioned now you haven't had a chance to follow immunologically, is there any evidence that you're seeing recognition of the tumor epitopes and not the virus in that potentially long-term immunologically mediated?
Yes. Well, the best evidence we have is the fact that when we re-challenge the animals, we re-challenge them with parental, naive, uninfected tumor cells, right? So yeah, we haven't examined exactly what tumor antigens or might be recognized. We are, however, taking a different strategy where we're actually introducing immunodominant epitope into the tumor, into the virus, which can be displayed on the viral envelope. And because this is, again, the problem with a lot of immunotherapy is that tumor antigens are self-antigens, so they're kind of weak antigens. And so here we've actually have preliminary data, which is actually going to be a thesis for my last remaining UCLA student, showing that you can insert an immunodominant epitope into the virus. It'll stably propagate that throughout the tumor and display a neoantigen, which then you can now target, challenge, with either neutralizing antibodies or antibodies targeting that epitope, or with CTL targeting that epitope, and that actually is quite.
The other route to use some immune checkpoint modulators,
Yes.
the CTLA4, CD1, or PD-L1
Yes.
modulator, so that you didn't not have to have strong epitopes.
Yeah.
You could actually do
Yeah.
better immunodominant epitopes.
Yeah. Well, the combination may also
Yeah,
be
the
quite good. Yeah.
combination would be a
Yeah.
very
Yeah. That's great. Thank you. Yes. Yes.
Any of the patients producing secondary tumors?
I'm sorry?
Any
No, we haven't. I mean, they're still, the follow-up time has been about 3.5 years now, but we've seen no evidence of any kind of secondary tumors. No. Thanks. Yes.
Thank you for the interesting talk. I have a
Thank you.
question to the intravenous injection agent.
Yes.
Because usually the most gene therapies have the limitation and specificity to
Yes.
get only into the tumor. How did you solve this problem actually?
Well, I think, in fact, it's solved for us by the tumor neovasculature. So it has been shown that there's what's called an enhanced permeability and retention effect. For any particles that are floating through the circulation, they get trapped by the tumor neovasculature. They leak into the tumor through the leaky neovasculature, and then they tend to get trapped there. Now, it's not an efficient process. And I think we need the difference between our previous studies in the research lab using 10 to the 6 titer virus versus the Tokagen studies using 10 to the 9 titer virus, which worked much better. I think indicate that we are getting a lot of trapping in the reticuloendothelial system and wasting a lot of virus in the circulation. But whatever does get to the tumor takes hold and replicates selectively, and that's why we can get this selective transduction effect.
And
Yes.
so far you don't see any side effect based on the virus infection intravenous?
No. So far, we've had no indication of that in animal models or in the patients. Yeah. Thank you. Thank you very much.